Hi, my name is Deirdre Mason. I am with the Association of State Drinking Water Administrators, and I want to thank you all for attending our webinar today entitled USGS Drinking Water Related Research, Data, and Tools. ASDWA's mission is to protect public health and the economy through the provision of safe drinking water. Our members are the Drinking Water Program Administrators from the 50 states, territories, the Navajo Nation, and the District of Columbia, and they regulate, provide technical assistance and funding for public water systems, and also coordinate with multiple partners. This is the first webinar in our series with the U.S. Geological Survey that will be ongoing throughout the rest of this year. So stay tuned for announcements about our upcoming webinars on a number of topics, including harmful algal blooms, water quality data, models and applications, long-term changes in the quality of the nation's streams, rivers, and groundwater, and understanding chemical and microbial contaminants in public drinking water. So for today's webinar, USGS will provide us with a broad overview of high visibility efforts related to monitoring data, tools, and products from the USGS National Water Quality Program and the Toxic Substances, Hydrology, and Contaminant Biology programs. Our presenters today are Patty Tocolino, and she is the Deputy Program Coordinator for the USGS National Water Quality Program. Her expertise lies at the interfaces between environmental chemistry, toxicology, risk assessment, and the fate and transport of contaminants in the environment. Patty is filling in today for Mike Facazio, who is the Program Coordinator for the USGS Toxic Substances, Hydrology, and Contaminant Biology programs. Next, we'll have Ken Balitz, who is the Chief for NAQUA Groundwater Studies. He has nearly 40 years of experience as a research hydrologist and scientific leader, including the design and implementation of large-scale comprehensive groundwater monitoring and assessment programs. And we'll hear from Neil Dabrowski, who is the Water Resources Assessment Branch Chief for the USGS Water Mission Area and Chief for NAQUA Surface Water Studies. He has been with the Geological Survey for over 30 years, studying water chemistry and designing and supervising large-scale multidisciplinary water quality investigations. Please note that a video recording of today's webinar will be made available for viewing on ASDWA's website within the next few days. And excuse me, the link there is wrong. Just go to www.asdwa.org. So if you want to watch the webinar again or have any people that are going to be not able to view the webinar today, they can come back and watch it later. And please type your questions into the box on your webinar control panel. We will not be taking verbal questions, but you may type your questions in at any time during the webinar, and we'll wait to answer all of the questions after all the presenters are finished speaking. So with that, let me go ahead and turn it over to Patty Tocolino, who will start us off. Patty, are you ready?
I am ready. Thank you, Deirdre. And thanks for the opportunity to speak with you all today. We're really excited to have this kickoff webinar and to be speaking with you over the next series of months. Today, we're excited to provide a broad overview of some of our high visibility efforts related to monitoring and data, monitoring and tools from the USGS National Water Quality Program. And we'll also give you a sneak preview of efforts by the Toxic Substances, Hydrology, and Contaminant Biology programs that Mike Facazio heads up, and those will be highlighted in future webinars. So USGS drinking water activities generally fall into two broad categories, and without diving into the details of the USGS organizational structure, we'll just talk about these in general terms here. So we've got basically a water quality and quantity side of things and an environmental health side of things. And the water quality part is handled by the National Water Quality Program that I'm part of, and also Neil and Ken are part of. And then there's the environmental health part that includes the toxic substances, hydrology, and contaminant biology programs that Mike Ficasio heads up. And there's a lot of interplay between these two areas, which we'll demonstrate as we go through all of this. On the water quality side of things, there's also a quantity aspect that's largely handled through our water availability and use science program. The water quality assessments that we'll be talking about today occur at the local, state, and national scales. And there's 4 main goals to the National Water Quality Program. We assess the status and trends of the nation's freshwater resources. So basically, we're looking at what's the current status of water quality, and is it getting better or worse? We also study the effects of human and natural factors such as land use, water use, climate change, to better understand water quality and ecosystem conditions. And last week, we forecast how water quality conditions will change in the future and how they're affected by things like human activities and management strategies. So the environmental health side of things, there's a lot of process-oriented exposure and health-related science activities. And some examples of those are the development of a whole variety of advanced analytical methods for things like pharmaceuticals, personal care products, pesticides and pesticide adjuvants, cyanotoxins, and a whole variety of bioassays. They also work to understand the transport of chemical and microbial hazards throughout the environment. And as I mentioned earlier, there's a lot of the water and the environmental health activities tend to complement each other. For example, the exposure-related environmental health efforts help to place the water quality findings into context, and water quality can take some of the analytical methods developed by the environmental health group and apply them nationally. Some examples of current and future environmental health work that will be highlighted in subsequent webinars are provided here. So some of these examples include exposure-related work, such as quantifying contaminant exposures in drinking water pathways, infrastructure, whether we're talking about water treatment, conveyance, or plumbing, can directly or indirectly lead to changes in the chemical or microbial content of water at the tap. And there are new study plans and a possible workshop co-sponsored by USGS and ASDWA that'll be talked about later. This work with human health experts is a new research effort underway, and it involves folks from the Toxic Substances Hydrology Program along with the National Water Quality Program. But more importantly, it includes human health experts from CDC, EPA, NIEHS, and some academic institutions to explore associations between drinking water exposures and human health outcomes. So we're really excited about that new venture. And the de facto water reuse in drinking water refers to situations where you've got a drinking water intake that's downstream from wastewater effluent. So pretty much a lot of places in the country right now. And this is most important where wastewater discharges account for a substantial part of base flow. And there's a collaborative effort underway right now between EPA and USGS on this topic. So stay tuned for more on these topics in subsequent webinars. The focus of today's talk will be on water quality, and we'll focus on some recent high visibility efforts related to drinking water that we're hoping will be of particular interest to ASDWA members. So we've got a few categories of information we'll be talking about highlighted here in these blue boxes. So the last thing I'm going to talk about here before I hand this over to Neil is give you a broad picture of the monitoring approach taken by the National Water Quality Program. And Ken and Neil will go into more specifics about our approaches for surface water and groundwater. But in general, we collect data from ambient, that's untreated, groundwater and surface water samples collected across large areas of the United States. And because we use nationally consistent methods for sampling and analysis and have low detection levels, that allows us to compare water quality conditions among broad geographic areas across the United States. And so with that kind of high-level overview, what I'm going to do now is turn it over to Neil, who's going to focus on surface water-related efforts, and then he'll hand it over to Ken, who will focus on some groundwater-related efforts. And so with that, Neil, I'll hand this over to you.
Thank you, Patty. Hopefully we're all seeing my screen now with the title slide. And I'm gonna...
Neil, you need to switch to the PowerPoint. We're seeing your other screen.
Oh, can you initiate the sharing again?
Just in the top of the control panel, you'll see a large pause button. Under that is a drop down, and you should be able to select it from there.
The showing application? How about that? Are we good?
No, actually, no. Read me off the options that you see in the drop down. Sorry.
Off the show application?
Yes.
PowerPoint slideshow, USGS PowerPoint application.
Hit the PowerPoint slideshow.
Okay, that's where I thought I was initially. Maybe it just hadn't updated.
Can you lift your PowerPoint into view? Seems like it's off the screen or something. It's wrong.
Still there. Hang on just a moment.
Ah, that's it. Perfect.
That's it? Okay. Not sure why. Okay. Thank you all. Sorry about that brief technical problem. Okay. Well, as Patty said, there's a number of water quality components being looked at in the U.S. Geological Survey, and I'm going to focus on two pieces of that. The National Water Quality Assessment Project findings, and that's because these are national in scope, and also some of the Cooperative Matching Fund program findings, which are fund local and state scale investigations. And just very briefly, excuse me, the right-hand column under goals has a shorthand for the goals that Patty had already reviewed. That is that we look at the current status of water quality. We evaluate trends, that is whether it's getting better or worse. We seek to understand the effects of human and natural factors. And then we want to forecast water quality conditions to see how they'll change and what the effects of human activities, climate change, and management will be in the future. The monitoring approaches that we use in surface water are varied, and they vary in scale and intensity. The most traditional is the National Fixed Site Network, and we collect discrete samples at a set of key locations throughout the year. We also conduct continuous monitoring at selected fixed sites, and we have sensors for a variety of parameters and constituents. And then we have the regional stream quality assessments, which is designed to identify stressors that impair aquatic ecosystems, but also to provide information on the spatial distribution of a much greater number of contaminants than we can measure at the fixed sites because the contaminants the lab expenses are quite high. And each of these regional assessments looks at 100 sites in a geographic area, but for a much smaller window of time. And then finally, we use all this data and more to develop models. And in particular, I'll share modeling results for nutrients and pesticides. And as Patty said, I'll be walking through some of our monitoring data, and then illustrating some of the products that we derive from the monitoring, modeling that results as well, and then some of the tools to share that data that you can access. So first, the National Fixed Site Network. The objectives of the Fixed Site Network are to characterize the status and trends of water quality and aquatic ecosystems. And we're doing that by collecting discrete samples of surface water and analyzing for nutrients, major ions, parameters including pH, DO, and temperature, dissolved solids, salinity, hardness, and taste, and over 200 pesticides. And a new addition is cyanobacteria toxins. The network includes fixed sites on large rivers and both coastal and inland, a number of reference sites, and a small number of small ag or urban streams that are indicators of chemistry in those land use settings. The sites are sampled yearly, and we usually collect 10 to 24 samples, and there is a website that serves this data that's updated annually. And this is the website for that data, the National Water Quality Network, water quality tracking website. There's 117 stream river sites in the network that are served through the site. And the site provides information on not just concentration, but also calculations of loads and yields for nutrients, pesticides, sediment, and also ecological conditions in the network. The first window on the left allows you to select individual sites, and the second serves loads and yields for major watersheds in the Mississippi River Basin, and the one on the right does the similar data for coastal rivers. As an example, we could dive into that first panel and get a summary for an individual site in the upper Midwest. And examples of the data, types of data you can access are summary of pesticide concentrations in comparison to human health benchmarks, not just for the current year, which here is shown as 16, but also for previous data stored in the database, in this case ranging from 1992 all the way up to 2015. The bottom right, you can also download any specific constituent, in this case nitrate, and show a time series for the entire period of record, in this case going back to 1997, as well as the current year served here, again, 2016 in the darker green. This current network is admittedly sparse, especially with respect to small streams. So a few years back, we launched an effort to utilize data collected by other entities at all levels, regional, state, local, and tribal. And we refer to this as the national data aggregation. And the data aggregation was a significant amount of work. We literally had a team polling agencies and entities across the country. But the result was both outstanding and also had some cause for concern. So when we evaluated the records, this example on the slide is the data we retrieved for nutrients. And what we found was that although we were able to get water quality data from over 600 monitoring organizations, we found that there were extensive interoperability issues arising from inconsistent approaches in documenting common metadata elements, or in short, much of it was unusable due to missing data. So for example, for the nutrient data, of the 25 million original nutrient records, over half of it, 14 million, had missing or ambiguous information for one or more key metadata elements. But we're learning important lessons from this, and US EPA initiated an interagency work group to improve the quality of metadata, and the National Water Quality Monitoring Council is establishing a work group to increase data submissions to the water quality portal using consistent set of metadata standards, which we really think is great because we plan to repeat this national data aggregation for data to include data through 2017 in the near future. So We did also, as I mentioned, have a massive positive outcome, and we retrieved over 185 million water quality records for 480,000 sites from 600 organizations, enabling the largest ever evaluation of trends in U.S. streams between 1972 and 2012. This enabled us to calculate over 20,000 trend results. These are trends for 51 chemicals and 38 measures of aquatic life, including nutrients, pesticides, sediment, carbon, salinity, and ecology. And there were 1,400 sites nationwide with at least one trend result. All of these trend results are included in the publication listed at the bottom by Gretchen Olsner. And these results will be the foundations now for answering critical questions about the causes and effects of these changes in stream quality. And the data is being, a subset of this data is being served online through a set of interactive maps. This is one example of interactive map, a screenshot from the Trends Mapper that can be accessed at the website shown at the bottom. The tool was released in 2017 with about 50 constituents served, including nutrients, pesticides, sediment, carbon, salinity, and ecology. And for this example, I selected a map of trends in chloride. And the sites that have a high probability of increases are in red, and sites that have a high probability of decreasing trends are in black. Our team has been analyzing the trends in chloride as well as sulfate from a number of stations around the country, because we can use this information to gain insight into the potential corrosivity of streams and rivers in the nation. In particular, the chloride-sulfate ratio is an important indicator of potential corrosivity in surface water, and as the ratio of chloride to sulfate increases, the potential for corrosion increases. And what we see in this pie chart is the area in blue in the bottom quadrant are sites across the nation where we have an increasing chloride to sulfate ratio, indicating that the potential for corrosion has increased from 1992 through 2012. I'd like to note that you just probably see the broad pattern of increasing chloride in red dots in the northern part of the country, often attributed to use of road salt. And there's also a trend of black dots in the northeast that is decreasing sulfate in stream sites. That's probably due to improvements in improvements in air quality, that is atmospheric deposition of sulfate, and resulting improvements in stream chemistry as a result of that. Okay, my screen is not advancing now.
Try clicking with your mouse on the little button in the lower right portion of the window.
Thank you. Okay, so continuing on corrosivity. So the potential of corrosivity shown across different landscapes on the left-hand panel in this figure. And what we found is that in urban waters in particular, the chloride to sulfate ratio, which is plotted on the y-axis, is much larger than in other landscapes. And again, we think this may be because of the use of road salt in urban environments. In addition, we looked at the seasonality of a select number of sites nationwide. And again, we have the chloride sulfate ratio on the y-axis, in this case, the months of the year on the x-axis. And we can see that the highest potential for corrosion occurred in the winter at this site and decreased, and then had a secondary peak in the summer. Again, this is most likely due to road salt use in the winter, and the summer secondary peak is probably due to low flow conditions in which there might be a higher proportion of groundwater input, which tends to be more saline, or maybe a component of wastewater. Basically, this indicates that simply monitoring quarterly might not be sufficient to catch some of these seasonal signals, and better stream and river monitoring could help optimize corrosion control in systems. Finally, we found that stream and river corrosivity was linked to increased probability of lead action level exceedances reported by drinking water facilities in the watersheds upstream. That is, that there's a statistical relationship between the occurrence of elevated lead at the drinking water facilities and elevated chloride sulfate ratios. So this plot has the chloride sulfate ratio on the x-axis and the probability of a lead action level exceedance on the y-axis. And we see that when the chloride sulfate ratio is less than 1, the probability of a lead action level exceedance is generally less than 50% at upstream sites. On the other hand, when the chloride sulfate ratio is greater than 3, the probability of a lead action level exceedance is generally greater than 90%. We're actually surprised that this worked out that well, and nonetheless, we see that the 95% confidence interval is quite broad, and this just indicates that there's a high degree of variability in this relationship. Another water quality issue that's received increased attention in the last few years have been harmful algal blooms and the associated algal toxins. This is traditionally thought of as a lake and reservoir problem, but in 2015 we had a massive bloom on the Ohio River, which impacted a large number of water purveyors. And this was a headline from an Ohio newspaper with a toxic algal bloom stretching 650 miles along the Ohio River. This occurrence made it abundantly clear that blooms can occur on large rivers, but there had been no national or regional assessments of cyanotoxins in the nation's large rivers. This motivated us to launch a pilot study during the summer of 2017, collecting HAB-related data at 11 large rivers in our national water quality network. We selected sites that have drinking water intakes somewhere proximal, either upstream or near downstream to the site. The pilot study objectives were to describe cyanotoxin occurrence in these large rivers during the summer of 17, and to assess the potential for harmful algal bloom occurrence in the nation's large rivers, using a combination of traditional and emerging approaches, including algal pigment sensors. In addition to just measuring toxins at these sites, we also collected data on algal community composition, chlorophyll, cyanobacterial genetics, and the sites that were selected were geographically diverse. Preference was given to sites that were also equipped with continuous water quality monitors for DO, pH, and algal pigments. And indeed, 7 of these 11 sites had continuous sensors for DO, and 4 had sensors for algal pigments and nitrate. As Deidre mentioned at the launch, we're going to have a seminar later in this series on findings from these studies and other HAB studies at the USGS. So I don't want to go into this in great detail, but very preliminary findings showed that the genetic screens showed that there was indeed genetic ability in the algal communities at this site for toxin production. Now, just because the genes that produce toxins were present doesn't mean that they're active at the time, but it does mean that there's a potential, if environmental conditions become favorable, for toxin production. So again, staff from Kansas Water Science Center, Jennifer Graham, will be speaking on this in the future. Now, in addition to the discrete samples at large rivers and some other studies nationwide, researchers have been looking for ways to use continuous water quality sensors to detect and better predict HABs. At the Kansas Water Science Center, researchers have been studying HABs in a reservoir that's used for recreation and drinking water. and looking at the use of continuous sensors that measure chlorophyll in real time to help predict when water quality problems would occur. While the USGS, in collaboration with numerous partners, supports a national network of hundreds of temperature and other water quality sensors, the network only includes about 40 sites in 13 states that are measuring levels of chlorophyll. These sensors, though, show great promise as forecasting tools for harmful algal blooms. And one example of this is the work that they did in Kansas at Cheney Reservoir. And the USGS Water Science Center staff there developed a relationship that looked at toxins as a function of chlorophyll measured using the continuous sensors and discrete samples for toxin concentrations. So the relationship that they developed is shown as this black line in the plot. That's the predicted. And measured data are the red dots, and the y-axis is the probability of exceeding 0.1 milligram per liter of microcystin. And you can see that the predicted relationship is pretty solid, accurately predicting the seasonal pattern over 2 months for the occurrence of microcystin. So this forecast would allow proactive rather than reactive management. And currently 3 drinking water sites in Kansas have operational real-time forecasts. And ongoing studies in other states such as Ohio have also had promising results. Similar spectral data could also be collected remotely by satellite, though I believe the resolution is about 30 meters, so although it will be applicable to lakes and reservoirs, we may be a little while before we can adapt results to large rivers. The USGS is participating in the CYAN project along with NASA, NOAA, and EPA to develop an early warning indicator system using historical and current satellite data, as well as ground-based discrete measurements of algal concentrations, that's chlorophyll, and toxin occurrence. And hopefully the findings of this project will support federal, state, and local partners in their monitoring efforts to assess water quality and predict blooms to protect human and aquatic life. Just as the algal blooms in large rivers had been somewhat unexpected, until recently, little is known about HABs and toxin occurrence in small streams. So as I mentioned, one of the national water quality assessment monitoring approaches were regional stream quality assessments. This is intensive studies of small streams, and we conducted this in these 5 regions from 2013 through 2017. In each of these regions, 85 to 100 streams were sampled intensively through the spring and summer to characterize the spatial distribution of pesticides, pharmaceuticals, mercury, nutrients, sediment chemistry, stream ecology, and including algal toxins. And the toxin work was done in collaboration with the Environmental Health Mission area of the USGS, the Toxics program there. The results shown here are for the Southeast study that we conducted, and we found that microcystins occurred in 39% of the small stream sites sampled in the Southeast US. Maximum concentrations were only 3 micrograms per liter. So it's not so much that these are a health risk, but the occurrence raises questions about the connection of small streams, lakes, reservoirs, and large rivers. And we hope that the data for these various components can eventually be integrated to get a better understanding of how toxic algal blooms evolve in freshwater systems. Finally, another bit of findings from the RISCWA samples. As I mentioned, the RISCWA was a means to cover a larger chunk of space in this country in a variety of settings than our 117 fixed sites. So what we found in the RISCWA sampling, and these are these are very preliminary findings, is that pesticides rarely exceed human health benchmarks. We had developed a new pesticide schedule for the studies in this cycle of NAQUA beginning 5 years ago. That includes a bunch of new high interest and more heavily used pesticides, particularly fungicides, and new analyses for degradates. At the 445 sites sampled by ASDWA, only 6 of the sites had a mean concentration above a human health benchmark, and 5 of these were for atrazine and were located in the Midwest, which is a known issue. Individual sample concentrations exceeded human health benchmarks 113 times in only 107 samples at 50 of the sites, and only 8 pesticides. And this is out of 4,000 samples collected during the studies. And again, most of these exceedances were for atrazine and were from the Midwest. I would like to mention that in these small agricultural streams, often we found 16 to 38 compounds in any one sample, and the effects of complex mixtures of pesticides are much less well understood than single chemicals. But again, recall, these are small agricultural streams and are never used as drinking water supplies, but are more just an indicator of what is present in the environment. I mentioned at the top that we used data from all of our studies to construct models, because no matter what your resources are, you simply cannot sample the entire resource in the nation. You have to use models to extrapolate in space and in time to identify sources and better understand the systems so that you can make predictions and forecast. So these models support decisions such as setting monitoring priorities, approving pesticide registration, determining how and where to spend resources on managing issues, and predicting potential effects. The model up on the screen is the WARP model, that is watershed regressions for pesticides, and the results shown are for atrazine probability that the annual mean concentration will exceed the human health benchmark of 3 micrograms per liter. The website also serves predictions for the probability of exceeding aquatic life benchmarks for fish, invertebrates, and fish. And in addition, it serves results for about 100 pesticides and covers 5 windows of time in 5-year increments from 92 through 2012. But again, the WARP model predicts concentrations in streams that the USGS monitored, and not for lakes and reservoirs. Another modeling tool that's well known is the USGS Sparrow modeling approach, which has been applied to a number of constituents, but has been widely used to predict nutrient loads and yields and identify sources of nutrients down to the level of stream reaches. Several of the last round of Sparrow models are available as online mappers on the USGS National Water Quality Assessment page, including a mapper for the Great Lakes region, a mapper for the Mississippi watershed, the Red Assiniboine River Basin, the Yellowstone River, and a Great Lakes mapper that I'll just show a quick screenshot from. This is a zoom to the watersheds contributing nutrients, in this case, total phosphorus yield loads to Lake Erie. And in addition to plotting loads for subwatersheds to the water body, we can also show zoom to select an individual watershed, and for that reach, show the proportions of loading from different sources, including point sources, urban land, manure, fertilizer, and natural sources in forests and wetlands for that catchment area. Although there's no drinking water standards for phosphorus and nitrate, it's only an issue in parts of the country. This nonetheless could be a tool for source water protection, since, again, elevated nutrients result in issues including problematic things like taste and odor issues in drinking water, in addition to algal blooms and toxins potentially. Then finally, to close, just share with you briefly some other tools for data discovery that the USGS has. This is the Water Quality Portal, and it's a cooperative service that developed by the USGS, the Environmental Protection Agency, and the National Water Quality Monitoring Council. It serves data collected by over 400 state, tribal, and local agencies, and also supports other applications that some of the other groups use, including EPA data discovery, EPA's nitrogen and phosphorus data access tool, and Delaware Water Quality Portal. And the screenshot just shows the entry point, and you can dive in. And once you're in it, you can look at the national results, and there are tabs that instruct you on how to use the tool and how to download data. This slide shows another USGS effort, the Human Health Benchmark page. And because many constituents that we measure don't have federal drinking water standards, since 1998, we've been partnering with the EPA and other agencies to develop health-based screening levels, HBSLs, to supplement existing benchmarks and place our water quality data in a human health context. These HBSLs are calculated using the same methodologies as EPA chronic human health benchmarks for pesticides, and we're well into an update of the website to account for updated toxicity data and updated methods, which should be completed within the next year. The update will result in a database with more than 804 contaminants, with 174 having HBSLs that we've calculated. And in addition to presenting the HBSLs, the website is intended to provide kind of one-stop shopping by including all of the most relevant recent EPA benchmarks. There's a number of sites that I won't go into any detailed description of, but USGS National Water Quality Program homepage also has a USGS sediment data portal for national data on sediment concentrations in rivers and streams, the National Atmospheric Deposition Program site that presents the chemistry of precipitation, the biodata, which has aquatic bioassessment data, and a page that presents estimated annual pesticide agricultural use. So in summary, USGS develops information relevant to drinking water quality for streams and rivers that tracks current water quality concerns while seeking to understand emerging issues. We collect discrete, synoptic, continuous, and remotely sensed data for sources and public water supplies. We develop models to predict concentrations and identify contaminant sources. And we serve online a number of tools for data discovery that that allow you to examine changes in water quality over time, to place the occurrence in human health context, and display model results. Thank you. I think we'll take questions at the end. I'd like now to pass the screen to the next presenter, who will be Ken Belitz.
Good afternoon. Hopefully you folks are seeing my screen. Neil, is it displayed?
Looks good here.
Great, thank you. So far you've heard from Patty, who is speaking on behalf of herself and Mike Focasio and Neil Dabrowski next. My name is Ken Belitz. And this afternoon, I'd like to focus primarily on results for groundwater collected by the NAWQA program. I'll share some other information with you. And again, as a reminder that Patty introduced, NAWQA focuses on the status of water quality in the United States. Trends, is water quality getting better or worse? Understanding, why might we see high concentrations? Why might concentrations be changing or not changing? And what you just saw, we're also now doing some forecasting as well. Let's focus a little bit on our assessment component, and then I'll discuss a little bit about understanding and trends. We are currently assessing groundwater quality in 3 dimensions. We're achieving that through the collection of data in 3 network types. One of our network types are land use studies. Another is major aquifer studies, and the third are principal aquifer studies. And the thickness that you see in each of those map images is roughly proportional to the depths. The land use studies typically are based upon data collected at observation wells, often 20 to 50 feet beneath land surface. From 2013 to 2022, we'll sample about 1,400 wells. If you look closely, you'll see green dots. Those are agricultural land use wells. Red dots are urban land use wells. In many cases, we have been sampling those networks since the 1990s. We have our major aquifer studies. They typically consist of domestic wells. In some of our more urbanized parts of the nation, they consist of public supply wells. Typically, these wells are deeper than our observation wells, 50 to about 150 feet. In this 10-year period that we're currently in, we'll sample about 1,200 wells. The land use studies, the major aquifer studies, were begun in the 1990s. Starting in 2013, we began sampling public supply wells as part of something we call principal aquifer studies. These wells are typically much deeper, 150 to 750 feet deep. We'll sample about 1,500 wells in this 10-year period. And then if you look at California, you'll see a very large density of wells. Those are wells sampled as part of our cooperative water program. In California, we have something called the Groundwater Ambient Monitoring and Assessment Program. The dots outside of California are the wells that we're sampling as part of our current 10-year cycle. NAWQA samples for a very large suite of analytes. We have water quality constituents with benchmarks. Geologic sources include things like trace elements, radionuclides, nitrate in some cases. A lot of the constituents that we sample for are sourced from human activity, nutrients, volatile organic compounds, pesticides and pesticide degradants, microbial constituents. We're also sampling for nuisance constituents, iron, manganese, dissolved solids, hardness, etc. Manganese has a secondary maximum contaminant level, but there's also a human health benchmark that we can use to put our concentrations into context. Many of the water quality constituents that we sample for don't have benchmarks, particularly the volatile organic compounds, the pesticides, and their degradants. Nevertheless, we have that data. We also sample for what can be called new analytes or analytes of emerging concern. In this 10-year period, it includes pharmaceutical compounds, hormones, polonium, enterococci, and others. That's the water quality side of what we do, and then there's the understanding side of what we do. We sample for a wide range of geochemical indicators, major ions, redox, pH, DO, temperature. We also sample for a broad suite of tracers of groundwater age. It includes tritium, in some cases SF6 or chlorofluorocarbons. We sample routinely for carbon-14, and we are sampling for noble gases as well. The noble gases allow us to get at both very young ages and very old ages. NAWQA is known for using its low-level detection methods. For VOCs and pesticides, concentrations typically below parts per billion. Although these concentrations might not affect human health, they do provide an indication of the human fingerprint. When we find these compounds in the subsurface, we have some sense that this water has in fact been in contact with the landscape. I would like to emphasize, as you've heard several times, NAWQA samples untreated water before it goes into the system. So this is a source water assessment. The primary means by which we are organizing our data at this time are principal aquifers. Principal aquifers are large geologic units that provide ample quantities of water for use. The U.S. Geological Survey has identified 67 principal aquifers in the United States, 20 of which provide most of our water supply. The 20 shaded regions that you see in this map provide about 90% of pumping for public supply and about 85% of pumping for domestic supply. So we're sampling those shaded regions in this 10-year window. When we sample an area, whether it's for a principal aquifer survey or a land use survey, or what we used to call a major aquifer survey, we use an equal area approach. The area of the aquifer system is divided up into cells of equal area. And you'll notice they're not squares. So, in some places they're fairly rectangular. So if you look in that eastern portion, and other places more equant. There are a number of wells in any one of those cells that one could sample. We pick one. Thus, within a cell, it's randomized sampling, but this is spatially distributed randomized sampling, and as such is stratified. When one looks at this, one winds up with a dispersed set of points across the landscape, where each area of aquifer has an equal opportunity to provide a sample. When you take that ensemble of samples together, you can begin to infer water quality for the whole. This slide shows, sort of tilted in the third dimension, our principal aquifer studies. There are various colored dots across that map. There are about 800 wells in 9 principal aquifers. We have fully processed all this information. We've collected the data. We've had it analyzed. It made it into data series reports, and then we presented some of our results in fact sheets, and I'll share those results with you today. Shown in gray are about 700 additional wells, most of which have already been sampled. Those results are still coming back to us from various laboratories. Those data will be presented in additional data series reports and fact sheets, so those are in progress. Today I'll share results with you from those 800 wells. And how does NAQUA characterize groundwater quality at a regional scale? The primary metric that we'll use today is proportion of the resource with high, moderate, and low concentrations. This might seem trivial or not, but proportion is scale invariant. We can talk about a very small aquifer, or we can talk about a very large aquifer, and our estimate of the proportion that is, say, high is dependent not on the size of that aquifer, but on how many samples we collected. So the confidence of an estimate depends upon the number of samples, not on the size of the area. So what do we mean by high, moderate, and low? If a concentration is above a human health benchmark, we consider that to be a high concentration. The demarcation between low and moderate depends upon whether we're dealing with an inorganic or organic constituent. If it's an organic constituent, we set the boundary between low and moderate as 1/10 of a benchmark. If it's an inorganic constituent, we set the boundary between low and moderate at 1/2 the benchmark. And the reason we've separated the inorganic and the organic is because organic constituents are generally introduced by people. There's a greater sensitivity to their presence in groundwater. Inorganic constituents, in contrast, occur naturally. They can be introduced by people, but typically they are naturally occurring. So again, proportion is our metric. It's scale invariant. It depends upon the number of samples with respect to how much confidence we have in that estimate. So let's begin, again, looking at this nice tilted map of the US. And let's begin by looking at the basin and range shown in green. We have 2 pie charts, one for inorganic, the other organic. You can see that about 20% of the basin and range samples, or 20% of the cells that we sampled, or about 20% of the resource have high concentrations, about half have moderate concentrations, and about 1/3 have low concentrations. In contrast, when we look at the organic constituents, there are no high occurrences and there's a small sliver of moderate concentrations. What we're going to do next is take that inorganic pie chart and break it into 3 components: trace elements, radioactivity, and nitrate. If you look at the yellow under the inorganic pie, you'll see the trace elements account for that entire slice. If you look at radioactivity in the basin and range, you don't see any high concentrations. If you look at nitrate, there are no high occurrences in the basin and range samples. Given that there are 60 samples, it's certainly possible we missed some concentrations that are high. Just based on the number of samples at a 90% confidence, one can say that nitrate is no higher than 3 or 4% high in that system. So in the basin and range, we see trace elements are the inorganic constituent class that account for the high concentrations. Let's take a look at the organic pie. What we see is a small, thin green slice for VOCs. Pesticide compounds do occur. They, in fact, occur quite frequently, but they occur frequently at very low concentrations, not exceeding 1/10 of a human health benchmark. Please keep in mind that a very large number of pesticides and their degradants don't have a health-based benchmark to put our data into context. So use the basin and range as a basis for looking at some other areas in the U.S. So again, here's our tilted nation. We're showing our 9 principal aquifers for which we're looking at data today, the basin and range in green. Let's bring in the Rio Grande just to the east of the basin and range. It's that purplish area through New Mexico. And you can see the pie chart is comparable. The inorganic constituents are present at high concentrations in more than a quarter, moderate plus high in more than half, and pesticides and VOCs do not occur even at moderate concentrations in those 60 samples. So it could be that up to 3 or 4% of the resource has high concentrations and we missed it because we didn't collect enough samples, but it would be in that range. Let's bring in 3 more of our study areas. Shown in green are the coastal lowlands belts around the Gulf Coast. Shown in red are the southeastern coastal plain, curving through the southeast. And then shown in blue is the North Atlantic coastal plain, which is North Carolina up through New England. Each of these principal aquifers consists of Cretaceous to tertiary sands and sandstones. And again, if you just look at the inorganic pie chart compared to the organic pie chart, you see a pattern that we saw in the basin range in the Rio Grande. We do see high concentrations. We do see moderate concentrations of the inorganics. But on the organic side, we generally don't see those compounds. The North Atlantic coastal plain is an exception. We see a small sliver in the 1% to 2% range for the organic compounds. Let's bring in the glacial deposits. The glacial deposits are this brown area extending across the northern tier of the United States. What we see there is about half of the samples, about half the cells, about half the resource have either high or moderate concentrations for inorganics. We did not detect either VOCs or pesticides at either high or moderate concentrations. Let's bring in one more, and then we'll discuss a little bit more. The valley and ridge are carbonates located just here along the Appalachians. Again, we see a pattern in these carbonate aquifers comparable to what we saw in the other locations. Inorganic constituents show up at high concentrations, high plus moderate in the 25% range. We see organic compounds present only at moderate concentrations, mostly at low. If you were to take each of these inorganic pie charts and pull them apart as we did for the basin and range, you'd find that trace elements account for most of that yellow slice. Radioactivity accounts for a small part of that yellow slice, and nitrate hardly accounts for the high values at all. This is the depth zone used for public supply. It's relatively deep, so perhaps that nitrate has not yet reached those larger depths. So let's now take a look at 2 more of our principal aquifers. They're a little bit different than the ones that we are looking at here. The Cambrian Ordovician principal aquifer is this pink-brown shaded region in the central northern Midwest. The Piedmont Blue Ridge crystalline is this large green belt that includes the Appalachians. And again, when we look at the story inorganic versus organic, we see comparable results. Inorganic constituents present at high concentrations, organic compounds not. Moderate concentrations of the organics do occur, but not as prevalent as the inorganic. So what I want to do now is take that inorganic pie chart, and for both of these 2 principal aquifers, we're going to break it up into 3 parts. We'll break it up into trace elements, radioactivity, and nitrate. The Piedmont Blue Ridge crystalline pie charts follow the same labeling as the ones to the left. In the 7 principal aquifers that we just looked at, trace elements were the constituent class that primarily accounted for the high concentrations. These 2 principal aquifers are different in that radioactivity accounts for the high concentrations. We can take a closer look at the Cambrian Ordovician today, and you'll see that this is radium. For the Piedmont Blue Ridge crystalline, it's largely radon. Radon doesn't have a regulatory threshold, it has a recommended threshold. Before we get to the Cambrian Ordovician, our results are available. 5 of our principal aquifers the samples were collected in 2013. It takes some time for all that data to come back to us, and then get that data into publications. We have a data series report that you can obtain all the data that we've collected, and then the results that we just shared are available in 5 fact sheets. In the following year, 4 more principal aquifers move through the system so that we can share the data with you through data reports and through fact sheets. They're all available on the web. And in addition, the Geological Survey now maintains a archival quality resource called ScienceBase. All the data found in these reports are stored in our ScienceBase. Now let's switch to the Cambrian Ordovician. So we'll kind of move from that status piece, what's out there, what's high, which constituents are prevalent at high concentrations, which constituents are not prevalent at high concentrations. In the Cambrian Ordovician aquifer system, we saw radioactivity was high. What you're looking at here are 60 public supply wells. They're distributed across the landscape using our stratified randomized sampling design. In addition, 20 samples or 20 public supply wells were selected because they were in areas of high gross alpha activity. Our work was done in collaboration with the Minnesota Department of Health and other states. Minnesota was particularly active in this work. The colors that you see then, blue are total radium activity less than 2.5 picocuries per liter, green 2.5 to 5, and then yellow greater than 5. The same 3 colors that we've been using previously, yellow high, green moderate, blue low. And then when you look at the aquifer system, you'll see a white area that's regionally unconfined, and then the gray area regionally confined, so that there are shales and other low permeability materials separating the Cambrian Ordovician from the overlying landscape. Let's start by looking at the unconfined portion. 22% of the samples, 22% of the cells, we can infer 22% of the resource has high concentrations for combined radium, 15% moderate, so about a third total. When we go to the confined portion, we have some striking numbers. More than half of the samples, more than half the cells, we can infer that more than half the resource has high concentrations for radium in that portion of the system. Only 9% of the samples are low. So this is an area of high concentrations for radium. Let's take a look at some of the underlying factors that can help us understand better what's happening. You're looking at a map. The circles are the proportion of the sample, are the samples with respect to their carbon-14 measurements. Any sample that has more than half the modern value of carbon-14 is shaded in a large red circle. Everything in a small yellow circle means that the carbon-14 is less than 5% of its modern values. You decay to that number in about 30,000 years. So what we typically see is in the unconfined portion of the resource, the groundwater is typically within 30,000 years old, and in the confined portion, the groundwater is more than 30,000 years old. Now let's look at the same locations. This time we're going to use helium-4 accumulation. Helium-4 is a good mechanism by which we can look at old groundwater. The yellow circles are less than 1,000 years old. The yellow-orange, 1,000 to 10,000. The orange are 10,000 to 100,000. The brown are 100,000 to 500,000. And then the red is more than 500,000 years old. When we look at the unconfined portion of the system, we typically see water in that 1 to 10,000 year category. There are some older samples. When we look at the confined portion, we generally see water more than 10,000, indeed more than 100,000 years old. And there's some samples that are 500,000 years or more. Well, this is a good surrogate, and we've looked at a number of indicators. As you move from the unconfined into the confined portion of the system, the water becomes increasingly mineralized, and it becomes increasingly reduced. And that matters, again, because if we look at the radium concentrations total, we see relatively low values in the unconfined young portion of the system. We see relatively high radium in the highly mineralized, relatively reduced portion of the system. When one looks at a number of lines of evidence, what one finds is that radium-226 is sorbed on iron hydroxide coatings under oxic conditions. But as the groundwater becomes increasingly reduced, those iron hydroxides begin to dissolve, thus mobilizing that adsorbed radium-226. As that iron hydroxide coating dissolves, there's less absorption capacity in the aquifer system. And as the groundwater becomes more mineralized, there's more competition for those absorption sites, and the radium is outcompeted by other ions. So we have increased competitive exchange. Those things all add up to radium-226 being high in the confined portion of the system. There's a set of other really interesting results, including radium-224 results. That work has been published in Applied Geochemistry by Paul Stackelberg and others, and it was published in Applied Geochemistry. Let's take a look at another topic that has drawn some concern. The production of unconventional oil and gas resources in the United States has focused concern on a number of things, including methane. We have been routinely collecting methane at a large number of our sites. The dots here are about 1,000 samples. The open circles are less than 0.001 milligrams per liter, the green up to 1, the gold 1 to 10, and then the red greater than 10. The 10 milligrams per liter is a benchmark for explosive potential, and we're using that just as a general indicator. So right away, when you look at the Western United States, you'll see that the values are generally less than 1. When you go to the southeast, one begins to see a preponderance of some of these higher concentrations. We've done some work looking at whether this is biogenic or thermogenic, and in nearly all cases, the methane is naturally occurring biogenic sources. This work was also published in Applied Geochemistry. Pete McMahon is our senior author, and you can take a look for that. That's just a very small sampling of some of the things that we've published or will soon be publishing. We have a number of studies looking at groundwater quality and unconventional oil and gas production. If you put Pete McMahon's name into a Google search and unconventional oil and gas, you'll find those. We have a paper pending on lead solubility indices for U.S. groundwater. The chloride sulfate mass ratio is a useful surrogate because it tells you something about the electrochemical potential of the water. We've been using some computational approaches to, in essence, titrating groundwater samples that are stored in our databases with lead to see how much lead can that sample absorb before it begins to precipitate it out. We have been routinely sampling our supply wells for hormones and pharmaceutical compounds, and that paper will be submitted shortly. Manganese is a constituent that routinely is examined through the lens as a nuisance constituent. It does have a human health benchmark, and we're evaluating manganese concentrations from that perspective. And there are numerous other constituents that we're looking at because they either have high concentrations or they are useful for better understanding water quality conditions. Not only are we using data collected by NAQUA, but we're using data from other sources. The U.S. Geological Survey has a national water information system. When our cooperative studies collect water quality data, they enter it into the system and then is available. For example, when we published our work on groundwater corrosivity, we used data from 22,000 sites, and that work was presented to this group about a year ago. Well, in addition, there are data collected, through folks like yourselves that allow us to paint a more complete picture of groundwater quality in the nation. There are about 150,000 public supply wells that we have precisely located in their XY location. And what you're looking at here are color codes. The gray locations are public supply wells, which on a two-dimensional map plot in a principal aquifer. In many cases, our principal aquifers are vertically stacked, so the two-dimensional location doesn't tell you exactly which principal aquifer it's in. Shown in gold are public supply wells that on a map are located outside of a principal aquifer. There's no principal aquifer mapped at all in the subsurface, and there are no glacial deposits present. So those gold locations are samples that we can now place into context because we have identified secondary hydrogeologic regions. There are about 15,000 of those wells that we can now bring into our analysis. Shown in gold are public supply wells that are located in areas that on a map are not mapped as principal aquifers, but in the deep subsurface there are principal aquifers present. Shown in blue are wells that plot outside a principal aquifer, but glacial deposits are present. And then shown in brown are locations where there's glacial deposits and there is a deep buried principal aquifer. We spent the past couple of years taking those data that are blue and brown and gray and pulling them apart so that we know what aquifer system or which secondary hydrogeologic region we're in. So we're about poised now to pull those 150,000 wells into the analysis. So that's the status side. We did a little bit of the understanding side. Now let's take a look at trends. And let's just simply ask the question, how does NAQUA assess trends? First and foremost, we have decadal resampling of our major aquifer survey and land use survey networks. Beginning in 2013, we began a new study type called Enhanced Trend Networks, and we're trying to understand the timescales at which groundwater quality changes that are more frequent than once per 10 years. At these sites, we have continuous monitors. We're looking at daily median values as a way of summarizing this continuous monitoring data. We have been sampling half of our networks once every 2 months for a period of 3 to 4 years, and then we swap over to annual sampling for 6 to 7 years. And when we do that inverse, we take the remaining sites and put them on a more frequent basis. The hallmark of much of what we're doing in this cycle is looking at age dating. So we have our decadal resampling. It helps us understand water quality changes to once per 10 years, enhanced trend networks, timescales shorter than 10 years. And we're also looking at what we call vertical flow paths. We've been sampling our land use surveys, relatively shallow, our deeper MAS surveys, and then still deeper PAS surveys. And when those are all pulled together, in essence, we're looking at the downward movement of water, and that downward movement typically occurs at timescales greater than 10 years. So we're looking to understand groundwater quality changes at timescales greater than 10 years through what we call vertical flow path studies. Today we'll focus just on this decadal resampling. We have a website shown here highlighted in blue that allows a person to look at changes in concentrations through time. We had a network or a series of networks sampled in the 90s. Many of those networks were resampled in the aughts, and they're being resampled again in the 10s. Our website allows you to compare and contrast network scale changes from each of those cycles. Our networks typically consist of 20 to 30 wells. We're using a Wilcoxon-Pratt sign rank test to identify changes at the network scale. I like to focus on the portion of the website here which shows inorganic and organic constituents. If you have that inorganic dialog box clicked, and then you just click on the downward pointing arrow next to the word uranium, you get a list of constituents. These inorganic constituents in this list are those that exceed a benchmark in more than 1% of our networks, which are MAS and LUS networks. If you then click on the organic dialog box, you get a list of organic constituents. Our organic constituents don't occur at high concentrations in excess of 1% of our sites. Instead, for the organic constituents, we're looking at the 5 most frequently detected VOCs and the 5 most frequently detected pesticide compounds or their degradants. So here you're looking at uranium, and you'll see that, for example, there are orange arrows that are small, there are orange arrows that are large. A small arrow indicates that the median of all the differences is less than 5% of the benchmark. So again, we're using benchmarks to indicate something that's small or large. The large arrow indicates that the median of all the differences is more than 5% of the benchmark. So consider that we're sampling once every 10 years, it may be 50 years before you begin to see things approaching a benchmark. In this case, the benchmark for uranium is 30 micrograms per liter, 5% is 1.5 micrograms per liter. You'll notice in the western United States, there's a large number of upward pointing orange arrows. When you look at the rest of the nation, you don't see those upward increases. Those large upward increases are largely related to changes in bicarbonate. When we irrigate in the arid west, we are putting plants into the ground. In the plant root zone, we're generating CO2. That CO2 is combining with that downward moving irrigation water. There's a downward moving bicarbonate front. That downward moving bicarbonate front is liberating uranium from the mineral surfaces. Again, this is a paper that we've published. Our lead author is Karen Burrow. And again, it's uranium and bicarbonate that are driving these changes. So that's a little bit on the understanding side. Now we'd like to move back to the status side. We're going to look at status. What is the quality of the nation's groundwater? And we'd like to know that groundwater quality, not just at a set of points, but we'd like to be able to map it wall to wall and top to bottom. We're going to be mapping constituents of concern, and we're going to be mapping at regional and national levels. And we're going to be relating those constituents of concern to factors that help us map it. And so we're mapping master variables, pH, redox, and groundwater age in order to help us map constituents of concern. The primary tool by which we're doing this is machine learning. The two primary mechanisms, the two primary machine learning tools that we're using are random forest and boosted regression tree. These machine learning methods are capable of handling dozens, even scores of potential explanatory factors, but they are data intensive. You can think of them really as pattern recognition, and the more patterns that you have available to put forward as an explanatory factor, the better your chances are of discovering the relationships. To do that, you also need a lot of outcome variables, hence the need to start pulling in tens of thousands of data points across the country in order to help find those relationships. Let's take a look at one example. We have several thousand wells available in California's Central Valley. And the image you're looking at here is a set of stacked images of the dissolved oxygen probability. So this is, what is the probability of encountering dissolved oxygen at less than 1 milligram per liter? Red means a very high probability of finding dissolved oxygen, such low values. Blue meaning a very small probability. So for example, what you see is the Western San Joaquin Valley. Things are relatively reduced. The Eastern San Joaquin Valley, things are relatively oxic. And as you head downward into the system, it becomes increasingly reduced. Well, now that we have, well, actually, before I go to the next image, let me give credit where credit is due. Our lead author on this work is Celia Rosecrans. This work was published in the Journal of Hydrology. So we have this three-dimensional distribution of redox conditions. We can use that variable along with many other variables to map something like nitrate. What we're looking at then here is nitrate at 2 depth zones. We've mapped nitrate fully in 3 dimensions, but we're focusing here on these 2 depth zones. Domestic wells at a depth of about 180 feet, public supply wells at a depth of about 400 feet. These brown areas are areas where the concentrations are relatively high. And you'll notice in the Eastern San Joaquin Valley, in this depth zone used to domestic supply, there are a fairly large proportion of the resource with high concentrations. However, if you were to integrate this entire domain, you'd find that nitrate occurs in about 5% of that domain at high concentrations when you look at this as a whole. If you focus only on a portion of the southeast San Joaquin Valley, then you can find the proportions start going up to 30, 40%. When we look at the depth zone used for public supply, we generally find relatively low concentrations of nitrate. It simply hasn't gotten that deep as of the present. This work was published by Katie Ransom and a large cohort group. Katie is a postdoctoral fellow at the University of California, Davis, and she's been working with support from the USGS. Let's look at where we're heading in the future. You just saw some of our results from the Central Valley. We're also working in the glacial system shown here in the northern tier of the United States. We're working in the North Atlantic coastal plain, and we're working in the Mississippi embayments. You'll notice by these dates, each one of these studies is starting 1 year after the previous one. So over a long period of time, we'll complete a large amount of intensive work. We have a large number of publications. We've shared some of them today with you. There are other publications we have not shared with you yet. We have more pending, and then we have still more planned. If we step for a moment outside of the NAQUA program, our cooperative water program also engages in water quality work. One of our larger studies is the Groundwater Ambient Monitoring and Assessment Program, or GAMA, in California. The GAMA program initially focused on deep aquifer assessments. They're presently focused on shallow aquifer assessments, and that work is sponsored by the California Water Boards. Let's take a closer look at the publications that came out of that work and are still coming out of that work. The data is available in data series. There are a set of fact sheets comparable to what you saw today, scientific investigation reports explaining those results, journal articles explaining those results, and there have been a number of geographic information system products as well. That's their website. I encourage you to visit. So let's summarize. NAQUA is assessing groundwater quality used for drinking supply. Human health benchmarks provide context. We're using data from multiple sources, the National Water Quality Assessment Program, or NAQUA, the National Water Information System, or NWIS, Safe Drinking Water Information System, or SIDWIS, and others. We're mapping in 3 dimensions based on data and modeling. We pursue an understanding, or we pursue research focusing only on constituents of concern. A constituent rises to the level of concern if concentrations are above health-based benchmarks, or if it's an emerging constituent. NAQUA samples literally for hundreds of constituents, and so we're selecting which constituents to pursue based upon one of those 2 criteria. We're also focusing on master variables such as pH, redox, and age, and a number of ancillary data sets, which we haven't discussed today, which allow us to do a better job of mapping. NAQUA data and information are available on the web. There are additional USGS groundwater quality investigations. We showed you one, but there are many others that would be worth hearing about. And that is the close. Thank you.
Thank you very much, Ken and Patty and Neil. And so if I can remind everybody to please type in your questions. So please type in your questions. I don't have any questions at this time, but I think that all of this information is great for state drinking water programs and for thinking about ways that they can use the USGS data. Here I have one question that just came in about what work is going on related to the PFAS, the per and polyfluoral alkyl substances research. Are you doing any of that at USGS?
I can take that question. Sorry. The answer is yes. We have several studies in our cooperative water program that are looking at various scales and looking at methods to collect those samples and reliably measure them. So it's at the early stages. Unless Patty or Neil, do you want to add to that?
No, I think that's a good summary. We're at the beginning stages, and our National Water Quality Lab is actively working on developing methods for detecting those compounds, a whole suite of those compounds at low levels. So stay tuned. It's something we're actively working on.
I think I would add, Patty, I believe that the Environmental Health Toxics Program has some research looking at that and at the persistence of those compounds in both source waters and their persistence through treatment.
Yes, thanks, Neil.
Can you just explain what you mean by scales in terms of what scale you're looking at those?
Sure. We have sites where the military had been fighting fires, and there's just sort of a plume that's being investigated. And then we're doing ambient background monitoring as well, so that it covers a large region, looking at basically what are the kind of the background ambient concentrations.
Okay, great, thank you. I noticed there's a little bit of correlation between some of the UCMR constituents and also the work that USGS is doing. You mentioned manganese and some of the cyanotoxins, and so with this PFAS research, it looks like you're coordinating some with EPA to look at those different constituents of concern. Maybe you could just talk a little bit more about how you're coordinating with EPA on those, or just including other kind of data sets. I know you mentioned SIDWIS, and maybe also you could mention the Clean Water Act, the water quality programs, GIS modeling data, that sort of thing.
That's a really great observation, Deirdre. So yes, we do work with EPA as they develop their UCMR program and their CCL program, their contaminant candidate list every 5 years. So we certainly share our occurrence data with EPA, and that's part of the information that they use to determine which contaminants to put on their UCMR and CCL lists. So there is some nice back and forth there. So yeah, so they're looking at both to determine whether a compound on the contaminant candidate list gets put up for a new regulatory determination. As you guys know, they're looking at both the toxicity of those compounds as well as the occurrence, because they need to be able to demonstrate that if they either change a regulation for a compound or add a new MCL for a new compound, that they have to be able to demonstrate that there's a demonstrable benefit to that, and that's partially where the occurrence information comes in. So there's a really nice collaboration there between the two agencies.
Great, thank you. And could you also, maybe we can share, I have a question about the address of the Human Health Benchmark Data website, but I'm not sure if we can pull that up at this time. We can share that with everyone after the webinar. And I can also just share just everybody's contact information. And this is where you can view the recording on ASDWA's website. Just as a reminder, you can send questions to Patty, Ken, or Neil, or myself, Deirdre Mason at ASDWA, and we'll be sure to share that Human Health Benchmark Data website. And I think we're at time, and we don't have any more questions, so I just want to thank you all again for speaking for our webinar today. I hope everybody stays tuned for the next webinars coming up on different topics that USGS will be presenting for us in future webinars throughout the rest of this year. And certainly if anyone has questions after the webinar, as I said, please send them along and we'll try to get those answered. So thanks again, everyone, for joining, and thanks for your time today. With that, I think we'll end the webinar.
Thank you very much, everybody, and for those that have stuck around to the end, thank you.